I'm really happy to see all the faces on the other side of the screen that I haven't seen in a while. And I hope that we can all get together again sometime in the near future. Hopefully this COVID thing will be done by this spring, who knows. But I wanted to say, first of all, happy Carnival. I was lucky enough to go to the Champions Parade, 2-3 years ago with the MSB conference. And with Emmanuel and, well, there was a lot of us that managed to stay up the entire night during this carnival thing, but it was awesome. And I know that you're definitely missing all the festivities today. And I appreciate you listening to my talk. I'm sure it's not really a good substitute for carnival, but I hope you find some things interesting from it. But one advantage of you being in Brazil is that you could be in Kansas, where it's now minus 28 degrees C. Our classes are canceled today because there's such a electricity loss that they're having rolling blackouts. So I think I looked this morning and it was 26 degrees in Brazil. So I'd much rather be there today than in this cold weather here. But for those of you that might be thinking about coming to Kansas who haven't been here before, it's not like this all the time. So you're welcome to come to Kansas. We'll just make sure you don't come in February. So what I thought I'd talk about today, Prakasi asked me to talk about some of the work that we've done in the area of separation-based sensors. So this is coupling microdialysis sampling with microchip or capillary electrophoresis separations, and then using different detection modes. And the idea here is to have a generic sampling system that you can then couple to a separation system that can determine multiple analytes in one run, and then use a detector that's selective for that specific class of analytes. And some of the applications that we're interested in are being able to make devices that could be used near animal for continuous monitoring in animal experiments. Also, we're interested in doing on-animal experiments to look at behavior and neurotransmitters, and also look at perhaps using some of these in the intensive care unit near patients so that you can monitor neurotransmitters in patients with epilepsy or traumatic brain injury. So a lot of you have heard this before, but for those that might be new to what we do in my lab, we use the microdialysis sampling as a technique as a sort of a generic sampling technique. So microdialysis sampling is nice because you implant a probe into a tissue of interest. It can be the brain, it can be skin, it can be liver or tissue. And basically the dialysis membrane, or the probe consists of a dialysis membrane that has a molecular weight cutoff. So only small molecules will pass through this probe. So basically what we do is we pump a perfusate that's similar in composition to the extracellular fluid that we're interrogating. And then small molecules that are in this extracellular fluid that aren't in the perfusate will diffuse based on their concentration gradient from areas of high concentration to areas of low concentration. The nice thing about microdialysis for a analytical chemist is that the samples that come out of the system are salty aqueous samples, and proteins are excluded. So we don't have to remove proteins or do centrifugation or any sort of protein precipitation with acid or alcohol. Also, because proteins are excluded, that means enzymes are also excluded, so we don't have to worry about enzymatic degradation of our samples. So microdialysis sampling is really an online sampling technique, but it's also an online sample preparation method. So usually how these experiments are done is the microdialysis sampling system is connected. The probe is inserted into the brain or other tissue of the animal, and the microdialysis perfusate is pumped through the probe. It collects analytes from the extracellular fluid of the tissue of interest, and then they're pumped to either an online injection valve or a fraction collector. And the other thing that's nice about microdialysis sampling as sort of a generic sampling technique is that there's a number of different types of probes that you can use. So the concentric cannula probe that I showed is the main probe that's used for looking at brain neurotransmitter release, but you can also sample blood. So you can look at transport across the blood-brain barrier by looking at brain and blood concentrations. And also you can use linear probes to look at soft tissues. So Craig, my husband, used this, for example, to look at differential metabolism of anti-cancer drugs in tumor versus healthy tissue. So all these probes, you can not only is it a generic sampling technique, but you can choose the specific probe for the type of system that you're looking at. This linear probe is also useful if you want to look at some sort of bioreactor system. So the differences here are this has a very small diameter because the brain is a very heterogeneous tissue. So we want to get it in a specific region of the brain, while the linear probe is more for homogeneous tissues like the liver or the skin, where there's not a lot of differentiation between one part of the tissue and another. So microdialysis sampling has a lot of other potential advantages. One thing is it acts like a synthetic blood vessel because there's really no loss of fluid. You can sample for long periods of time with no fluid loss because the sampling is based totally on diffusion across the microdialysis probe. Also, once you implant the probe in the animal, you can do microdialysis sampling on awake, freely moving animals. So the animals can run around in that bowl and you can look at their activity and feed them and give them water and just let them go about their business and then monitor the pharmacokinetic transmitters. As I mentioned, they're aqueous samples and protein-free, and the enzymes are also excluded. So that makes the sample prep much easier. You can also put more than one probe in an animal, so you can monitor both brain or a couple of different tissues at the same time. And also importantly, because you can continuously sample in a single animal, the animal serves as its own control. So typically for a pharmaceutical company to do, say, pharmacokinetic studies of a drug, they would take from a rat every so often, and you can only take I think 10% of the total blood volume of the rat. So that limits how often you can sample. So you can only get the pharmacokinetics from that certain number of points. In addition, if you're looking at drug metabolism and you want to look at how much of the drug gets into the brain or some other tissue, then you actually sacrifice animals at different time points and measure the concentration in the tissues at different times. With microdialysis, you don't have to do that. You can just continuously monitor in the same animal the tissue or blood concentrations. So an important thing about microdialysis is because it's the sampling is a diffusion-based method, the slower you pump the perfusate through the probe, the more analyte you get into the probe. So if you pump at a slow enough flow rate, you basically sort of reach equilibrium with the extracellular fluid. And so your recovery is close to 100% because the concentration of the analyte in the extracellular fluid is the same as that in the perfusate. So if you go down to flow rates of about 100 nanoliters per minute, your recovery becomes close to 100%. Now, if you increase the flow rate, you decrease the recovery, but you do actually get more mass into the, it's a more dilute sample, but you do actually get more analyte into the sample. But typical flow rates that are used are usually around 1 microliter per minute, 1 to 2 microliters per minute. So a typical commercial microdialysis system usually consists of a bowl where you can have the rat running around, the microdialysis syringe pump for the microdialysis perfusate, and then a fraction collector to collect the samples offline. So if you're perfusing at 1 microliter per minute, typically people would collect maybe 10 microliter samples for LC analysis. So like I said, the typical flow rate is 1 microliter per minute. So usually like for many neurochemistry experiments or drug experiments, collected like 15 minutes, so you get a 10 or 15 minute, 10 or 15 microliter sample to analyze. If you're doing, say, catecholamines, you usually have to add acid and maybe a thiol antioxidant to make sure that the sample won't degrade. With the offline analysis, it's easier to do multiple analysis because you can have multiple fraction collectors for the different probes. And you can also take, with offline analysis, you can take a single 15 microliter sample and analyze it by more than one technique. So typically people do LC, UV, EC, fluorescence, or mass spec. But if you use, say, capillary electrophoresis, where you require very little sample to inject, you can actually take that 10 microliter sample and run it on several different types of analysis systems. But in the offline analysis, the temporal resolution really depends on the sample volume requirements of the analytical method and the sensitivity and selectivity of the analytical method. So if it's got good limits of detection, you can use small samples and get the results. If the analyte is at really low concentrations, you may have to collect long periods of time to get enough analyte in your sample in order to be able to detect analytical system. So to give an example, I really like this example. It's sort of an old example, but of using microdialysis to look at drug disposition, so pharmacokinetics and pharmacodynamics. Here we had a rat in the bowl. We gave the rat Ritalin. We injected Ritalin, which is used for attention deficit disorder. And basically what you can see is that you can monitor, we have one probe in the brain of the rat and a second probe in the blood probe. And so first thing we can do is, if you look at the red dots here, the red dots are the methylphenidate or the Ritalin that's in the blood. So you can watch once you dose the rat at 0 time, you can watch the methylphenidate increase in the blood. Then, because we have a probe in the brain as well, we can also look at how much methylphenidate is getting into the brain. So that's these green squares here. And then, then we can also look at what is the methylphenidate doing to the brain chemistry. And you can actually watch the dopamine concentrations increase once the rat is exposed to methylphenidate. And the other thing is, if you look at these bars, this particular microdialysis setup has a light on it that will monitor how much the rat moves. So once the methylphenidate kicks in, you can see the rat starts moving around a lot more. And then as the blood levels decrease and the brain levels decrease, the activity of the rat decreases. So one of the big questions that sort of drove us to the separations-based sensor approach is temporal resolution in microdialysis. So one of the things when you're setting up an experiment is like, what kind of temporal resolution is needed? Most neurochemistry experiments, you want really fast temporal resolution. I mean, ideally, like Whiteman's group and the fast scan voltammetry can do things in milliseconds or seconds. But you want to be able to get as much information as quickly as possible. Pharmacokinetics is much slower. Drug disposition is usually not super fast. So there you don't need as fast a temporal resolution as neurochemical experiments. And then you might also use this for a bioreactor where you might be wanting to monitor just once every 15 minutes or so, or maybe once an hour. The other thing that affects the temporal resolution is the concentration of the analyte in the dialysis sample. So as I showed, the microdialysis probe recovery is dependent on the flow rate. And also it's dependent on the compound, how it partitions across that dialysis probe. So if I have really low concentrations in my dialysate sample, I'm going to have to collect for longer in order to be able to analyze it. Also, if we're doing drug analysis, if the analyte is protein bound, it won't cross the membrane. So some drugs are really highly protein bound, like 99% protein bound, and only 1% is free. The free drug is actually what causes the pharmacological response, but With microdialysis, you can only measure the free drug because the protein-bound drug will not pass through the membrane. The other thing that's really important to consider is detection limits and the sample volume requirements of the analytical method. So do you need to do derivatization and fluorescence detection in order to get down to nanomolar or picomolar detection limits? Or is the thing at such high concentrations that you can do UV detection? EChem, we can usually get down to about micromolar detection limits. And then mass spec has some of its own issues with ion suppression, but it's also been used with microdialysis sampling. The other thing is what's the sample volume required? Like LC normally requires 5 to 10 microliters. CE requires basically as much as you need to get the sample close to the CE interface. If you're injecting it online, it's only a few nanoliters. Biosensors is usually a flow-through type thing, so the sample volume would just be the volume of the detector. One thing with offline detection is that even though you might be able to analyze, say, a 1 microliter sample using capillary electrophoresis or some other technique, handling a 1 microliter sample reproducibly is difficult. And when you collect it, you can have problems with surface tension or evaporation that can give you irreproducible results. Now, we're going to focus today on online methods, and the temporal resolution is also dependent on how fast you can do the separation or the analysis. If the analysis takes 10 minutes and all the rest of this you can do with 1 minute samples, then your temporal resolution will still be 10 minutes. So one of the things that you want to look at is trying to make the method as selective as possible for the class of analytes that you're interested in, so that you're not waiting for stuff to come out that's being detected that you're not interested in. So this is an example of online microdialysis LCUV that Craig's group did many years ago. But here they had like a valve, and so they basically used the syringe pump to a uni-switch valve, and then continuously monitored, I think this was caffeine metabolism in a rat. So even with LC analysis, you can get good temporal resolution. Here, I think was every 30 seconds with the online system. Okay, but this was done. All these things that I just talked about were all done with a rat in a bowl, tethered with all this tubing and swivel and everything. And some of the problems with this is you have a delay time between when the sample is collected and when it actually gets to the analysis system. So you have a delay time, plus you can get band broadening of the peaks or the sample as it goes from the probe to the analysis system. And also the rat is freely moving. It can move around the bowl, but it can't really behave. I mean, it can't really do anything interesting or exciting, maybe eat, but that's about it. So there's a lot of disadvantages we felt of this awake animal tethered online analysis system. Like I said, the large dead volume and all the tubing limits the flow rates that you can use, because if you go to really low flow rates, it takes forever for the sample to get from the head of the rat to the analysis system. It also has limited temporal resolution because it depends on the concentration of the sample that you're getting and the sample volume requirements of the analytical method. So if you're using LC, you're probably going to need about a 5 or 10 microliter sample to go into the LC system. And you have long delay times if you go at low flow rates to get that high recovery. You have long delay times between the time you collect the sample and actually analyze it. And then, as I mentioned, although the animal is awake and freely moving, can eat and everything, it's difficult to perform any behavioral studies other than just measuring its activity. So that we started thinking about, well, can we do this with freely roaming animals? And I know some of you guys have seen this already, but This is one thing we did early on that I haven't brought up in a while because it's sort of old, but one of the first things we thought was, what if we just use an osmotic pump on a rat and just collect off on the rat? So we had an osmotic pump. These osmotic pumps are actually used for drug delivery to the brain, and they just run based on osmotic pressure. Basically, it's like a toothpaste tube where the osmosis causes a continuous flow rate through a hole in the device. And so we connected this. You actually implant this underneath the skin of the rat, and then we connected it to a probe that was placed on the rat, and then we just made a little thing with the collection vial. And of course, the problem with this is you have to collect off the vial, but the flow rates were pretty low. I think they were about 200 nanoliters per minute. So we were getting good recoveries. It was just taking a long time to get sample. And so this shows we injected these rats with L-DOPA and looked at how much of this L-DOPA got across the blood-brain barrier and what it was metabolized to. These are metabolites of L-DOPA. And you can see we're only getting, I think, like half-hour samples or even maybe hour samples here. But this rat could just wander around without being anything. The problem was we had to take the little vial off the head of the rat and then analyze those samples. So it was possible to monitor behavior while collecting the dialysate samples, but the temporal resolution was dependent on the collection time. So we had to stop and take the vial off whenever we felt we had enough volume. We did analyze this offline, and obviously taking the vial off the rat's head was sort of a pain, but it showed that you can do sort of on-animal analysis in a freely roaming animal. So then we started thinking, well, can we do on-animal analysis using microchips? And, you know, microchips allow you to miniaturize a lot of things. And we know we can do all sorts of microdialysis sampling, different probes, different tissues. So if we can customize our chip with a detector that is unique for a class of compounds, and then either integrate the detector or build a detector for that specific class of compounds, then we have a sensor that can continuously monitor a drug and its metabolites or several amino acid neurotransmitters in near real time on animal. Okay, so our strategy, which we've been working on for quite a while, was to take our microdialysis. Well, we had to develop a interface between the microdialysis probe and the chip. Then we needed to develop electrophoretic separations for the analytes we're interested in. In some cases, if we were doing fluorescence, we needed to also integrate a derivatization step. We needed a detection step. Also, if the animal's roaming, we need to miniaturize the electronics and to get the data. So the second part of this, the sensor, because the microdialysis, the probes are pretty set and it's a good sampling method, was microchip electrophoresis, which all of you know a lot about. But it had a lot of advantages for this specific application. A lot of our analytes are charged, so it separates based on charge to mass. It requires really small sample volumes, just like capillary electrophoresis can analyze, you know, sub-microliter or microliter samples. A big thing that we found that was really helpful was that you can do manipulation of fluids electrokinetically. So that makes it really easy to do it remotely because you can just tell the electrodes on a chip which ones to turn on and off to inject and to separate the analyte. I'll show that we can integrate forms of online sample prep. Of course, the fact that you can do fast separations, a minute or less, are also very small of the microchip electrophoresis really makes it possible to bring, as a lot of people in Brazil have done, experiment as opposed to I think that's really powerful because you can put the system right next to the rat or right next to the patient or on the animal, and continuously monitor what's going on without having to sort of take the rat to a mass spec or collect samples and then analyze them offline. So as Wendell commented, we do a lot of electrochemical detection, and we felt that electrochemical detection was really ideal for this application because a lot of the things we're interested in are electrochemically active. It's easy to miniaturize because you can use standard photolithography techniques to integrate detection electrodes into the chip. And we can easily make miniaturized detectors. We worked with Pinnacle Technologies, and they developed this miniaturized isolated potentiostat that we could use to detect used with in-channel microchip electrophoresis with electrochemical detection. So when we decided to try and combine the microdialysis to the microchip electrophoresis on animal, we used a chip that was designed earlier in our group by Brian Wynn. And this chip involves it's a normal simple T chip with a larger channel above where you can have the microdialysis sample go through there. So the microdialysis sample goes through this top part. And then as I'll show in a minute, we can use electrokinetic manipulation and gated injection to inject sample continuously into this separation channel. And so this particular device was all glass with a platinum electrode, and was used in some of our on-animal studies. So as I mentioned, the ways that you can actually inject microchip or microdialysis samples into microchip electrophoresis. This gated sample introduction we use because we can do it remotely, but there's also a pneumatic valve setup that Scott Martin developed with our group, which works really well and is especially good for cell analysis. And then Bob Kennedy's groups used droplets to collect samples. That's another possibility, although lots of times those are collected offline and then analyzed by mass spec, which is a very powerful tool as well. So there's many different approaches, but if we want to actually put it on animal, the gated sample introduction works the best. So here you can see the microdialysis sample goes through this top part. We have a high voltage that goes from we apply a high voltage here, so that forms a gate of buffer so that the microdialysis sample goes to this waste reservoir. But then when we float this potential, we get a small plug of analyte that then goes into the separation channel, and we reapply this, and we can separate that sample that we just injected. And then we can do the whole thing over again once we've got our data from this first injection. Just to show some early data, David Scott used this just to look at monitoring nitrite production. And he used one of Fricassi's favorite compounds when he was working in my group, DEA nanoate, although I think Fricassi liked proline nanoate better, to look at NO production. But what he was really looking at is the formation of nitrite from the NO. So NO degrades to nitrite. And so he used this to see if he could detect nitrite continuously in an in vitro type situation. And so here, this is just online monitoring of nitrite using that platinum electrode with the glass chip. And you can see it increases over time as the nitric oxide is produced and then converted to nitrite. So then we wanted to see, can we do this on animal? So the idea with on-animal analysis is attractive because the animal is awake and freely roaming. So if you want to do behavior studies, you can look at the behavior of one animal or multiple animals. They can go about their normal activities. Also, if you're able to put the device on animal, you can minimize the distance between the probe and the analytical system. So at least in theory, you can minimize the lag time between when you get a response and when you actually detect it. We're really interested in looking at neurotransmitters or drug levels and how they affect behavior. So this approach would allow us to monitor both brain and maybe blood concentrations of drugs. But we need to do this, we have to miniaturize everything and build in telemetry for remote data collection and instrumentation control. So we picked the sheep as our model system because even though we can make a lot of this small, we can't make it small enough to go on the back of a rat like we did the little rat hat thing that we had before. So basically the system, we picked the sheep because they are larger animals. The system would fit on the animal. They're relatively docile. They're pretty well behaved. And they also have a lot of social behavior. There's a lot of studies regarding social behavior. And actually in New Zealand now, there's quite a few studies regarding, there's an Alzheimer's model for sheep. So there's a lot of possibility of doing some of these studies in large animals. So the idea then was to take this system. So this is the whole microdialysis system. So this is the electrochemical detector, the high voltage power supply, the syringe pump, the chips up here. So take all of that and put it in this box. And so we worked with Pinnacle Technologies, and they developed a system for us. So we put our chip in here. It has a small power supply, integrated electrochemical detector with telemetry, and everything's done by remote control. So we have the isolated potentiostat in here, and we can continuously collect the data. So this shows how it's set up. So we just did at first a linear microdialysis probe. So we just put it under the skin of the sheep. So we just removed some of the wool on the sheep and put the probe underneath the skin. And we don't want to do any brain surgery on any animals, especially sheep, before we actually are 100% sure that we have this working really well. So here we just had it on the back of the sheep. And then we're continuously, what we did is we used nitroglycerin as a model analyte. Our group does a lot of work with nitrate and nitrite. So nitrite's a good model or detection analyte for us. So basically we took the nitroglycerin, which is metabolized in the skin, to nitric oxide, which can then produce nitrite. And we had this linear probe underneath the skin. So as the nitrite was being produced, it would be diffuse into the probe, and then we can detect it. And so here you see the nitroglycerin, and we can see there's a lag time. There's still quite a bit of a lag time, which we need to improve. But every 10 seconds, you can detect the amount of nitrite that's being produced by the nitroglycerin in the sheep. And then one of the things we wanted to do is compare it with offline analysis. So we actually put a separate probe on the other side of the sheep and just collected like we sort of did with the rat with just vials, and then analyze those by liquid chromatography. So you can see liquid chromatography does not give us as good a temporal resolution as the online separation space sensor, but you can see now this is two different sides of the sheep. So the general pattern's the same, but it's not actually the exact same spot on the sheep. So you don't expect them to be exactly identical. So the idea then is to correlate this neurochemistry with behavior. So our idea is to take this system and I'll show in a minute some of the work we're doing with catecholamines, but the idea would be to be able to use this in the brain of a sheep and monitor neurotransmitters while sheep are in a pen, say, freely roaming and socializing, and then be able to get information about neurotransmitters along while they're behaving. So toward that end, we started focusing on catecholamines, which are really what people are interested in looking at for behavior and neurodegenerative diseases. And one of the things that we've looked at a lot is L-DOPA, which is transported across the blood-brain barrier and forms dopamine, and then it forms some of these metabolites. The problem with catecholamines, or the challenge with catecholamines, is we did the on-animal work with metal electrodes with platinum because that worked fine for nitrite. But for catecholamine analysis, as Buzz Adams showed way back in the 70s, it's really the preferred, carbon is really the preferred electrode substrate. And so we found that carbon electrodes are not as stable with the high glass bonding. And so Rachel Saylor and my group looked at two alternatives, a PDMS chip or a PDMS glass hybrid. And there's two different types of electrodes we've looked at. One is putting a carbon fiber directly into the chip. The other is using pyrolyzed photoresist on a glass quartz chip and then using a PDMS channel on top. And so this shows the PDMS glass with a PPF electrode. So in this case, one of the problems that the group worked on that they solved was they would get delamination of, because you have pressure flow this way, you would get delamination of the PDMS from the glass substrate. And so they figured out a way to get around this so that you can have this PPF electrode in the channel and also have microdialysis flow without it delaminating. So Rachel developed this nice separation of these L-DOPA metabolites. And here you can see this is just an online monitoring of a solution of them. So you can see we can detect they're all separated and can be detected using this online microdialysis system. Then we evaluated this just taking a homogenized rat brain and putting L-DOPA in there. So this was just an in vitro characterization. We added nitrotyrosine as an internal standard just to, it had several roles. One was to look at the recovery across the probe, but also to make sure that the electrode wasn't fouling and that we were getting reproducible injections into the chip. And it's hard to see here, but you can see there's a lag time. Then you start seeing L-DOPA. And you do see a little bit of dopamine production in the after a while in the sample, and it's really slow. But you can see this is the L-DOPA that you see, and then you start seeing dopamine after about 10 minutes, and it sort of levels off after about 70 minutes. So then the next thing to do was to look at, see if we can see L-dopamine in vivo. And the hardest part of this, and one of the things we're struggling with right now and trying to improve, are the limits of detection for dopamine for these experiments. So Shamal and my group, along with Anton Beribin, and Sarah Thomas did an in vivo experiment with high potassium stimulation. So if you change the perfusate from artificial cerebral spinal fluid to a high potassium perfusion, that will stimulate the neurons to release dopamine. And so we can't at this point detect endogenous concentrations of dopamine, but when we do this experiment, we can actually see, here's a couple of, so he perfused the rat brain twice with potassium, and you can see that there's some dopamine release. And if you look, it's a really small peak. It's this X peak here, but it does increase. It's not there when we first perfuse, when we just do the normal perfusate, but when we stimulate with high potassium, we see this dopamine peak. And so this is what we're struggling with right now. We can't detect it, but if we do something to stimulate it, we can actually detect the dopamine. So what we're working on right now is trying to lower our LODs so we can detect endogenous levels and not just stimulated amounts of dopamine. But we're encouraged by this, that we can actually detect dopamine released in the brain by high potassium stimulation. So we're working on some other analytes, some serotonin analytes, metabolites, and other metabolites of DOPAC that we've added to the separation. And this shows a microchip separation of all of these. The whole separation takes about 2 minutes, but the actual separation of the analytes we're interested in is only about a minute. And so one of the things we want to look at is for HVA and DOPAC, which are major metabolites of dopamine and L-DOPA, and see if we can't detect dopamine itself, can we at least detect the metabolites that are being formed? So the other area that we've looked at is been looking at traumatic brain injury. And I talked about catecholamines, but Schmal in my group worked as well on adenosine metabolites. So adenosine's really involved in the bioenergetics of the brain. And when people have a traumatic brain injury, they will actually remove part of their skull to relieve the pressure. And then there are studies where people will put a microdialysis probe in the brain to measure glutamate or lactate, glucose. And so it is possible to actually monitor other analytes as well. So we're interested in adenosine because that's one of the compounds that's believed to be affected by traumatic brain injury and epilepsy. So here, instead of using a pyrolyzed photoresist electrode, Schmal used a carbon fiber electrode, so similar setup, but using carbon fiber instead of PPF. And he was able to separate out the adenosine metabolites. And this is an in vitro where we're looking at the production of inosine, which is a metabolite of adenosine by adenosine deamidase, which we can then follow every 2 minutes. And then the other thing we're still working on that. That again, that's another one where we're trying to get better limits of detection for in vivo studies. And the last thing that I was going to talk about, and I'll do this fairly quickly because I spent 15 minutes screwing around trying to get on this, but is looking at amino acids. So we're also interested in looking at glutamate, aspartate, other excitatory amino acids. And our favorite derivatizing agent is naphthalene dialdehyde, which will react with primary amines to form these CBI derivatives. The nice thing is this is not fluorescent, but in the presence of cyanide, these primary amines will form fluorescent derivatives. And a long time ago, we set up a chip where we had online derivatization on the chip. So we bring in the microdialysate. We have a sort of an NDA cyanide and microdialysis separation here, and then we mixing here, and then we do the separation here. And one of the things that we did that I thought was really interesting is we looked at aspartate and glutamate as well as fluorescein. So we looked at fluorescein across the blood-brain barrier, and then this is endogenous concentrations of aspartate and glutamate in the brain of a rat. And so it would be nice if we could also do this system with this online, like on-sheep, on-animal system. We're still a little bit of ways from that, but Nate Orbany and my group built a portable LIF detector that's a little bigger than the lunchbox type thing that we had for the EChem detection. But he developed this. This is just a simple chip that a lot of you are aware of, just a long 15 centimeter chip, sort of serpentine type chip. And so he used this detector to look at amino acids in the dialysate sample. So this is a spike sample, and this is an actual microdialysate sample separated using that chip and his detector. And then he worked, now he's working at Jet Propulsion Lab looking for life on other planets. But while he was still with me, he looked at COMSOL modeling to look at the kinetics of the derivatization and optimizing the mixer for this online derivatization of the NDA and cyanide. And he came up with this chip, which is part glass and part PDMS. And this just shows some data that he got looking at spiking with glutamate and then looking at the lag time and the for glutamate, which is one of the important neurotransmitters. So with that, I just hope I've shown you, like, we've made some progress on making these modular platforms for separations-based sensors. We still have work to do, especially with trying to get better sensitivity for some of these assays. But the idea is to have like unique chips for different sets of analytes that people would be interested in monitoring continuously, especially in the brain. So neurotransmitters, reactive nitrogen oxygen species, amino acid neurotransmitters, peptides. So we're really interested in having like specific chips that you can sort of plug and play for specific analytes of interest. And so our future directions is, I mean, our ultimate goal is to develop these modular on-animal or near-patient monitoring systems, but we need to, we're working on improving the detection limits for catecholaminesine. We're working on trying to figure out how to put a decoupler in there that would help. And then we're also looking at the LIF detection, trying to miniaturize that more so it could actually go on animal. And then eventually we want to apply these to behavioral studies, drug metabolism, and we have people that in traumatic brain injury and epilepsy that are interested in monitoring these eventually near patient. So there's a lot of people that worked on this, but Shamal here is one of the key people. Let's see, Anton and Galina have worked quite a bit on this. And then there's a lot of people that have worked on this that aren't on this slide. So they're all here. And of course, we have a whole nother area of research that on reactive oxygen and nitrogen species that a lot of people that are listening to this talk have contributed to. And I didn't talk about any of that work today, but I really appreciate all their efforts in that area. And we're making some progress there as well. So with that, I'd like to thank you, and I'd be happy to answer any questions. And actually, hopefully you're still there. I can't really tell. You still there?
Yeah, thank you too.
Okay.
Yes.
It's sort of funny. About halfway through, I'm thinking, oh, are they still talking? Or I'm just talking to myself.
Wendell, you want to control the question section?
Okay, you can. Okay, we have time for questions. Thank you, Sue. Very nice talk.
Oh, thanks.
If you don't want to ask the question personally, you can put the question on the chat.
Mm-hmm.
If you don't want to do in English, you can do this in Portuguese. We will try to translate as better as possible.
It'd be hard for me to answer it if it's in Portuguese.
No, Susan has to work her Portuguese out.
Yeah, I tried. I was listening to Duolingo for a while.
Really?
Yeah, but now I'm just trying to learn Spanish because Portuguese is too hard, I think.
Spanish is more useful there in US.
Yeah,
maybe if you go to Miami.
Right, but most of my favorite people are in Brazil, so I probably should learn Portuguese.
Okay, questions? Emmanuel.
Susan?
Yeah.
Susan, if you stop sharing the screen, then you can
see you guys? Okay.
Yeah, you can see them.
I have to figure out how to do that. Hold on.
There is somewhere stop sharing.
Oh, I think I figured it out. Okay, let's see.
Yeah, now you're presenting.
Yeah.
No.
No.
Oh, stop presenting.
Yeah.
Okay. Yeah, I got it. Okay.
Hello, Susan. Great to see you again and see all this amazing work you do on the little animals and seeing them running around. It's always great to see all that complexity that you deal with. It's really nice. Have you ever tried to model with how is the people from machine learning, mathematics coming along? You have someone that try to model behavior with the chemical analysis?
Oh, you mean, so we did have, we were working with Pinnacle. They were developing a video recording. So the idea was to do video recording along with the animal behavior. We haven't done that yet because we're still trying to get the systems to get to that concentration. As far as the modeling of like the console modeling of the chips, both Manjula and Nate have done some work with that. Nate, who's gone now, did mainly he was working on modeling the reaction kinetics and trying to make sure that the samples were reacting long enough and mixing well enough for the derivatization reaction so that we'd get the optimal yield in the chip. But we haven't done any sort of machine, to look at behavior and the animals. I don't think we've gotten far enough. I wish we were that far.
Yeah, because that will be the maybe next level. Once you have the analytical part settled, and then the machine learning and the artificial intelligence comes into can start to predict the model, and then you do experiments to see if they feed back to the system.
Right. Yeah, no, that would be good. Yeah, we want to do. The ultimate idea is to have video monitoring so that if you see a trigger in an increase in the neurotransmitter, let's say you're at home and it's just the animals are either in lab or in some pen somewhere, but you can remotely monitor what they're doing, then you can go back and look at the data and see that the dopamine levels went up and these two sheep were fighting, or they were eating, or the serotonin levels went up and they were eating a lot of hay that day or something like that. That would be the ideal thing, or vice versa. If you look at the videos and see some unusual behavior going, then you can, so then you could use the kinds of things you're talking about to start correlating behavior with specific neurotransmitters.
And just one short question, or window actually. For the Pinnacle guys, do they have any model of C4D detectors?
No, I don't think. No, they don't. Not that I know of. Yeah.
Okay.
Yeah.
Because they sound pretty smart.
Yeah. No, they. I mean, I think they're selling the isolated potentiostat because they built that, and then and they used it for, it's a modification of their in vivo biosensor system, but they've also made it's in their catalog a separate system that's just isolated potentiostat for electrochemical detection. But as far as I don't think they have any conductivity detection, but I don't know, Wendell, have they talked to you about it? Maybe they are interested in it. I don't know.
Thanks a lot.
Yeah, it's good to see you. Good to see you.
Get back to Wendell to coordinate the questions.
Okay, thanks, Emmanuel. Alberto, to the next.
Oh my God,
you,
what
Sue.
happened to your confetti?
I changed the carnival background, so I'm more calm now. So thank you for your presentation. Actually, it was a masterclass on microdialysis. So I think it was really, really complete. So thank you for this. Thank you so much. I think it's a strategy that we could use with more frequently here in Brazil. It sounds really, really interesting. And my question is, how far can we go on the miniaturization of these systems? Because it's not just a question of the detection, but also, for example, the reagents that you need for complete the channels and the microdialysis.
Yeah.
And what do you think? How far can we miniaturize these systems?
We can definitely make it smaller than it is right now because we were using a laptop battery. But yeah, I mean, what we had we used the syringe pump we used was one that they used for diabetic patients for insulin. And definitely some of the different parts could be, I mean, we had talked at one point about using maybe an electroosmotic pump or something like that, where we wouldn't have to worry about, a big syringe pump. And also if we went to slower flow rates, that would, you wouldn't need as much sampling. One of the things that you might be insinuating is, basically the waste reservoir obviously gets, you just sort of, I think when Dennis did his thing, he just let it sort of drip down the cheap side, right? The waste sample, but you could also collect that as a total amount and analyze it later. But as far as I don't think we can get it as small as to go on the back of a rat at this time, but I bet we could maybe do a large rabbit.
Do you think we can reach the size of a kind of a wristband for smaller animals?
I think I'd need to talk to Apple or something before I.
All right.
Yeah, I mean, I think you're right. We talked to Pinnacle about trying to figure out how to. You could definitely make the chip smaller and do like a circular type channel instead of. I mean, we use just our regular planar chips, but and the power supplies could definitely be smaller, and the batteries could definitely be smaller. But I don't know if you could get it small enough to be on the wrist. Maybe if you integrated I mean, if KU had, a major electronics fabrication area, it might be possible, because you could integrate a lot of things that we can't integrate. But I guess you're in charge of one there, so maybe I should come to Brazil and build it, right?
All right, thank you.
Yeah.
More question? Didn't see anyone. No, Susan, I am curious about the portable LIF system.
Mm-hmm.
How large is it?
How big is it?
Yeah.
It's about twice the size. Well, it's about, let's see, I'm trying to think what would be. I think it's about maybe a foot high and 8 inches deep, maybe. It's not super big, but it's not super small either.
Okay, it's medium. Okay.
Yeah.
Nice. Oh, Elton, please.
Hi, Elton.
Hi, Professor Susan.
How are you?
I'm good. I'm good. And you?
Good.
It's really nice. It's really nice to see you again.
Yeah, it's good to see everybody. I'll have to take a screenshot of everyone.
Yeah. So regarding the microchip with the realization step on chip, how it works? Because I remember that this step takes some time, right? Like around 10 minutes.
Yeah, it sort of depends on. I mean, that's what Nate was working on. Yeah, because I guess you were really familiar with that, right?
Yeah, I remember. Yes.
So he was working on that in terms of the console modeling, because if you get, I mean, he was trying to improve the mixing, and
okay.
Yeah. So, and also, it doesn't have to be 100% complete. It just has to be reproducible, right? So
That's right.
But I think it still takes a couple minutes for sure in the chip.
Okay. Okay. Yeah, makes sense. Yeah, thank you. Yes.
You're welcome. Thanks. Good to see you.
Yeah.
You're doing well?
Yeah.
Yeah.
And here, I'm in Sweden, like it's really cold as well.
Yeah.
It's snowing a lot.
Yeah, COVID's bad there, right?
Yeah.
Yeah.
Unfortunately.
Gazil, please.
Hello, Susan.
Hi, good to see you. How are your kids?
Nice talk.
Oh, thank you.
It's just a curiosity. Have you used 3D printing for this kind of research?
We've talked. I was in Tasmania for a while with Breadmore's group, and we were talking to them about developing a system with 3D printing, and Galina and my group was working with them until COVID hit, and it sort of has gone on the side. But I think 3D printing would help a lot in terms of just the connectors and stuff, if nothing else. The idea was to, like, you could put a glass, or you could put, just make everything easier, because you could put a glass chip in a holder, but you could hold everything together with the 3D printing, and then also the online derivatization. I think you could easily integrate with 3D printing, because the volumes are the channels are larger than going into the chip. So yeah, no, I think we have a couple of 3D printers here at KU, and we had a design that we were working on, and that just Galena was meeting with this person pretty regularly, and then COVID hit, and we haven't talked to him in a while.
Okay, nice to see you.
Yeah, very good to see you. Your family good?
Yes.
Good.
Elton, do you want to ask another question? Oh, no. Okay. Yeah. Ever one more? More questions? Your baby is here.
There's a toy there. Emmanuel, how is your daughter?
Oh, Susan, she's dating.
How can that be?
She's dating.
Oh no. You're seeing all these people with these little babies, and you're remembering, right?
Yeah, she's 16.
Oh wow.
And she's dating.
Oh my gosh. Wow.
Even the other day, she was putting all her little, like, sorting the toys, and she recovered the Jayhawk.
Yay. Yeah, I need to bring more next time. We need to make sure all Wendell's kids are outfitted in Jayhawks.
Yeah. Will be Jayhawk and Corinthians.
Right. Yeah.
Richard, please.
Yeah, just had a quick question. Have you looked into any electrode modifications to improve the LOD?
So yes, Shamal looked at, especially for the adenosine, he tried several different things. He tried some nanoparticles. He tried some diamond carbon electrodes, because they reduce fouling. And then for the catecholamines, we haven't done too much. We haven't really tried much with the catecholamines, but we have with some of the other compounds. And the other thing that would be interesting to do that we haven't worked on too much would be looking at modifying it for selective detection of thiols, like glutathione and glutathione disulfide, and we haven't done that yet either.
Okay, thanks.
Yeah, thanks. Stay warm.
Yeah, I'll try.
Yeah. Vladimir, please. Hey.
Hi, nice presentation. I like the lab on a chip. You know, we already use the wrong words. We call chip, chip, and so on. But I was wondering about the high voltage power supply on the animal. I was thinking, when we have, for example, a battery, if we have like 1.5 volts, it depends on we may have like plus 1.5 or minus 1.5, depending on which side we ground. Okay, so I was thinking, what would happen if the chip is on an isolation platform? So we have some part of the electronics at difference of potential is like 2,000 volts, but from the chip to the ground is floating potential. But at the exact moment when the chip is put from this isolated platform on the ground, I was thinking perhaps is experience some kind of electric shock.
Well, no, but I mean, it's a good question, because with the electrochemical detection, the ground is at the detection end, and the fluorescence is backward. but with when we did capillary electrophoresis connected to CE with electrochemical detection, we had to ground the joint between the microdialysis, and we actually sort of grounded the joint between the microdialysis sampling and the the CE system for that reason, so it didn't go back to the animal through all the fluid, because there's a lot of fluid in there too, right? We didn't see anything with the sheep. We saw a lot of noise before we put it in that Faraday cage. We didn't notice any. I mean, the whole thing is self-contained in a Faraday cage, but it is connected to the sheep through liquid, which is salty. So
Yeah.
it may be feeling something. I don't know. It didn't appear to, but it could be.
I was wondering perhaps if the ship is all the time with the feet on the ground, perhaps it will never feel nothing. But
yeah, it's always
on the isolated platform and then to the ground, perhaps it can feel something.
Yeah,
when we
did the experiments with the sheep, it was in a barn and they were just on a concrete floor. So and we didn't lift them up at all. But it's a good point, because it is more of a problem, I think, with the small animals, because it can go back. I mean, there's less body weight too. I mean, it's going to be more effective, and it could go back through the liquid to the animal, right? So we usually take a precaution where we ground the interface between the dialysate and the chip, so that it doesn't do that.
Okay.
Does that
Thank
make
you.
sense? Yeah. But it's a good point. It's definitely. I mean, can't remember how many volts that we put across there. I think you're right. It's about 2,000 volts. The currents aren't really high, which is another thing. The currents aren't high. So.
Thank you.
Yeah, thank you. We just won't let the sheep pick their feet up.
More questions? Yes, louder, please.
Oh, hi.
Yes.
Thank you, Susan, to giving us this nice talk. I think you can show us a lot of things that you will have a great experience, and share your experience with our INCTBio. So thank you very much for accepting to give this seminar.
Oh, thanks for inviting me. Yeah.
And I have just a question, a philosophy question. In your opinion, what is the bottleneck of this kind of microchip devices to be applied in real world?
I think for us, and I don't know if this is general or just us, but the ruggedness and reproducibility from chip to chip, being able to make a chip that, especially with the electrochemical detection, that behave. I mean, our students will make, or my students will make the microchips with the PPF electrodes, and half of them will work, the other half there's something wrong, or then like the glass bonding is better, but it's harder, more difficult, and PDMS is much easier to make like prototypes with, but it's not going to be good for a commercial. So I think the main thing is to get a design that is reproducible, integrated, and can be mass produced. So you almost have to make something that can eventually be done with injection molding or something, which is why doing 3D printing or we've also been working with Soper's group a little bit, trying to make some plastic chips so that to see if we can make something that you could eventually maybe, do with injection molding, so then you could make hundreds of them reproducibly, or thousands of them reproducibly. I think right now, we're still in that sort of science fair mode where we're making stuff and it doesn't always work. And I think being able to get to that point where you can transfer it to rugged design is probably the hardest thing.
Thank you. May I ask one question?
Sure.
In your opinion, the microdialysis membrane, it's possible to improve, or do you think that is already we reach the best material or best membrane to extract some neurotransmitter or some other analytical that is important for biological purpose?
No, I think there's a lot of improvement for the microdialysis that could also be done. One thing, Kennedy's group's been making some micro-fabricated probes or just to make smaller probes so that you can use them, like Mike Johnson at KU is doing some zebrafish experiments. It'd be really nice if we could have a really small probe that could go into a zebrafish brain. Or the other thing is, sometimes you don't want just small molecules. You might want a better recovery for peptides. That can be a problem. There's only 3 or 4 different kinds of membranes. I think having some membranes that would, like some of them are too hydrophobic, so hydrophobic compounds don't get across them and they have really low recoveries. So having membranes that give you better recovery for some of these hydrophobic compounds would be good. Yeah, I think there's a lot of things that could be done on the microdialysis side for sure. And there's only a few groups that are really trying. Scott Chippy's group in Chicago, he's got some really small capillary tubes for microdialysis sampling. And then there's also push-pull perfusion probes. But I think it's an area that could use a lot of additional membranes and different additional configurations. And then of course there's like what you do, the SPE that you can put directly in the brain too, right? And monitor like Paul, I can never say his last name right, but Balutzen. Anyways, from up in Canada, he's done some really nice work putting SPE probes in the brain. There, the temporal resolution will be dependent on taking the probes out. But if you could, one idea would be to, and people have done this, put things in the perfusate that shifts the equilibrium towards recovery of the analytes, so you get more out. So you can put things in the perfusate like antibodies or cyclodextrins or things like that that will force the equilibrium towards the perfusate so you get a better recovery. So there's a lot of things you can do that definitely help that. Sorry, that was a long answer for a short question.
Any more questions? So not sure if you check it, but Diogenes, Bruno, they are also in the room.
Oh, okay. Bruno, hello.
Hi, Sue. How are you?
Diogenes, yes.
Hi, Sue.
I think Katie
nice
sent you a letter pretty soon, right?
Right.
Yeah.
So, do you try and work it with the dual channel?
Yes.
For decrease
yes,
limit of detection.
I didn't talk about that, but yeah, Manjula and And a new student, Indica, have been using it for bipolar. So they've been, instead of doing the dual electrode, they're doing a bipolar electrode across both channels and then changing the electrochemical signal into a fluorescent signal. But it's a similar configuration to what you worked with in Kansas. So it's still there. Still useful, yeah. In fact, I talked about some of it at, well, I don't know if any of you are going to PitCon remotely, but Andy Ewing got the award and I gave a talk at his symposium. So I mentioned the dual electrode stuff in there. So.
Okay.
Good to see you.
Yeah. Good to see you. Good to see you.
Valeria, please.
Hi.
Hey, hi.
Hi. In fact, I don't have a question. I just wanted to say goodbye because I need to leave.
Really good to see you.
It was really good. Oh, so nice, your baby, Wendell. Baby, what's her name?
Laís.
Laís. Hi. So I have to leave. It was so nice to see you, Susan.
Yeah, really good to see you.
Even to see in online. Okay?
Right, yeah. It's like a family reunion.
Yeah, it's like family.
Yeah.
Well, I hope to see you face to face.
Me too. Yeah. Hopefully this won't last much longer.
Yeah. Bye.
Bye. Take care.
More questions?
Laurus, do you have a question?
No question.
So, if there are no more questions, I would like to thank you, Sue, again, for your very nice talk, and good to see you. And
yeah.
The same with all the Brazilian visitors. All of them were online.
You should send me your. Except you probably give your talks in Portuguese normally, right? I was thinking you should send me your list of speakers, but if they're all in Portuguese, maybe I won't listen.
Yes, Fracassi has the next one, maybe. So thank you so much. Fracassi, do you want to speak a little more?
Yeah, just to close the section. So I have to thank you again, Sue, for the outstanding presentation, Wendell for the handling the session, and also for the introduction. Also, we'd like to thank you, thank all the participants of this webinar, because we reached more than 60 people at the room. So it's really, it's being actually amazing, this kind of webinar of the National Institute of Science Technology, because we can group people from different parts of the country and also now outside the country. So this is really interesting. So thank you so much, everyone, for the participation. And Lauro, you want to say some final remarks?
Just to say thank you and congratulations for Susan for nice talk. I think we can learn a lot about the seminar and your experience. So thank you very much for teaching us a lot of things about microchips, microdialysis, bioanalysis. So we are learning a lot during this section. So thank you very much. And it's a great opportunity to the bioanalytical members to learn and to have some experience about the bioanalytical or bioanalysis things. And thank you very much for giving us this opportunity. So thank you very much. And I hope to see you soon, face to face.
Yeah, me too. I appreciate that. And thank you so much for inviting me. It's really good to see everyone, actually. And you know, we missed LACE last year, and it sounds like we might not have LACE. So hopefully we'll get together sometime soon, because I really miss seeing everybody once a year, and at least once a year. Yeah. And if you do somehow figure out how to get on this side of the ocean or this side of the world, make sure that you stop by Kansas. So, and I promise it won't be minus 26 unless you come next week.
I just forgot to mention, but Sue is international consultant of the National Institute of Bioanalytics. So thank you also for this presence at our institute. Thank you so much. And also,
for our institute.
Yeah, it's an honor. And also merchandising for the first time participants, that every month, once a month, we have these webinars. We are trying to keep this on Tuesdays at 4 p.m. Brazilian time. And on the first or the second week of the month. So we don't have an official channel for this, but we have a page on Facebook that we put the seminars there, the webinars there. And any question you can also send to us and at the institute. So if you don't have any more questions or comments, so we can close the session. Thank you again, Susan, for the for the
thank
presentation.
you for inviting me. Thank you. It was great. It's good to see everybody.
Thank you, Susan.
And I was lucky the power didn't go out while we were talking, so that's good.
Now we are going to the champagne section.
Yes. Yeah. I have to go get my beer.
Okay, guys. See you all, everyone. Hope so.
Bye-bye.
Next webinar.
Have a good one.
It's good.
Bye-bye.
Take care,
Bye-bye.
everybody.
Thank
Yes.
you, Sue. Thank you so much.
Thank you, Sue.
Thank
Bye,
you.
everybody.
Take care.
See you next
Bye,
month.
Diogenes. Bye, Laurel.
I will send you an email, Susan.
Okay. Email
If you
or
have some ideas.
I got your file now.
oh, you just got it? Well, it's a good thing that we figured out the Safari thing.
Nice.
Hey, Susan, I hope I can visit you one day.
Please, yeah, I know. We keep on trying to get you here, yeah.
But not in this time because I don't like cold weather.
No, this is not a good time to visit. The best time, I think, is the spring or the fall.
Yeah.
Although the summer, I guess, is sort of Brazilian, right? It's pretty
Too hot.
hot, yeah. So I don't think the summer bothered you, did it, Prakasi?
Oh, actually, when I just arrived there, it was hotter than in Brazil.
Oh, really?
Yeah.
August is not a good time to come, but